Hello everyone. Today we are going to learn about cell cycle. Cell cycle is the sequence of events take place in the cell which includes DNA replication and cell division from a single cell to two daughter cells. In the cell, this process takes place in a cyclic and sequential manner, so that it is called cell cycle. Cell cycle includes two main phases, M phase, that is mitosis phase, which lead to division of a cell, and the longest one, that is interphase, which includes G1 phase, G2 phase and synthesis phase, which leads to growth of cells. So, in this module we are going to learn about cell cycle and phases of cell cycle. Learning objective of this module are introduction to cell cycle, phases of a cell cycle checkpoints in general, because we are going to study this topic in the separate module. Duration of a cell cycle, the journey of a life that starts from a single cell to form a complex multicellular organism goes through numerous cell divisions. All organisms are made up of cells which constantly divide and multiply. Cells divide when they need to replace injured or dead cells, or as a part of the development of a fertilized egg. 300 million cells are replaced in our bodies every minute without our even noticing. Studies on yeast, worm, flies, frogs, mammals and plants have contributed to a universal picture on how the basic cell cycle machinery is regulated, and research on these many divergent organisms is also elucidating how evolution modified the basic cell cycle machinery to cope with the specific developmental and environmental changes of each organism. Do these division occurs randomly or are they regulated? Cell division follows a specific pattern called the cell cycle. This cell cycle illustrates the different phases of division. These divisions are regulated and follow a strict chain of events. Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, interphase, mitosis or nuclear division, and cytokinesis or a division of the cytoplasm, that is M phase. The basic cell cycle. The cell cycle is an ordered series of events leading to replication of cells. The two main events of cellular replication that represent the two larger phases of the cell cycle are the copying of cellular components, interphase, the cleavage of the cell, that is mitosis. During interphase, appropriate cellular components are copied before cell can enter mitosis. Cell has to pass through rigorous control known as checkpoints. Interphase is further divided into subphases as will be discussed in the next section. Mitosis, M phase, is the part of cell cycle when the cell prepares for and completes cell division. Chromosomes condensed during the prophase period of mitosis by tightly folding loops of 30 nm chromatin fiber attached to the chromosome scaffold. Sister chromatids produced by DNA replication during the S phase remain attached at the centromere and multiple points along their length and become aligned in the center of the cell during metaphase. During the anaphase portion of mitosis, sister chromatids separate and move to opposite poles of the mitotic apparatus or spindle. Segregation of the two sister chromatids to each daughter cells. Since the cell cycle is a cycle, it has no distinct beginning or ending. Cells are continually entering and exiting the various phases of a cell cycle. Figure illustrates the fate of a single parental chromosome throughout the eukaryotic cell cycle. Although chromosomes condense only during mitosis, they are shown in condensed form to emphasize the number of chromosomes at a different cell cycle stages. The nuclear envelope is not depicted. Following mitosis M, daughter cells contains 2n chromosomes in a diploid organisms and 1n chromosomes in haploid organisms, including yeast maintained in the haploid state. In proliferating cells, G1 is the period between birth of a cell following mitosis and the initiation of DNA synthesis, which marks the beginning of the S phase. At the end of the S phase, cells enters G2 containing twice the number of chromosomes as the G1 cells. The end of G2 is marked by onset of mitosis, during which numerous events leading to cell division occur. The G1, S and G2 phases are collectively referred to as interphase. The period between one mitosis and the next. Most non-proliferating cells in vertebrates leave the cell cycle in G1 and entering the G0 state. Phases of cell cycle. Interphase generally lasts at least 12 to 24 hours of mammalian tissue. During this period, the cell is constantly synthesizing RNA, producing protein and growing in size. By studying molecular events in a cells, scientists have determined that interphase can be divided into 4 steps: GAP1, that is G1, S, that is synthesis phase, G2 phase, that is GAP2 phase. We will begin our discussion of the events take place during interphase with those that occur immediately after a cell has successfully divided during mitosis. This phase is called G1 phase. Figure illustrates the phases of cell cycle and their relative duration. G1 typically the longest phase of a cell cycle. This can be explained by the fact G1 follows cell division in mitosis. G1 represents the first chance of new cells have to grow. Cells usually remain in G1 for about 10 hours of the 24 total hours of a cell cycle. The length of S phase varies according to the DNA that the particular cell contains. The rate of synthesis of DNA is fairly constant between cells and species. Usually, cells will take between 6 hours to complete S phase. G2 is shorter, lasting only 3 to 4 hours in most of the cell. In some, then interphase generally takes between 18 to 24 hours. Mitosis, during which the cell makes preparation for and complete cell division, only takes about 1 to 2 hours. GAP1 phase. G1 is an intermediate phase occupying the time between the end of cell division in mitosis and beginning of a DNA replication during S phase. During this time, the cell grows in preparation for DNA replication and certain intracellular components such as the centrosomes undergo replication. Before a cell begins DNA replication, it must ensure that it is biologically ready to take on such a process. Cell increases in a size in G1, produce RNA and synthesize protein. G1 is the phase when this cellular monitoring takes place. An important cell cycle control mechanism activated during this period ensures that everything is ready for DNA synthesis. During G1, the cell reviews the cellular environment and the cell size to ensure that the conditions are appropriate to support DNA replication. Not until the cell ready does it leave G1. If all is not ready to undergo DNA replication, cell can pause during G1 and enter a phase called G0 or G0 depending on a cell's preparedness to continue in the cell cycle. G0 can last days, weeks or even years. When the cell has reached an appropriate size and is in a supportive environment for DNA replication, it will exit either G1 or G0 and enter the next phase of interphase called S phase, gap 0 or G0 phase. There are times when a cell will leave cell cycle and quit dividing. This may be a temporary resting period or more permanent. Example, a cell that has reached an end stage of development and will no longer divide. Example, neuron cells. A G0 cell is often called quiescent. The term quiescent is used in respect to cell cycle activity and does in no way symbolize lack of metabolic activity. In fact, many G0 cells are terminally differentiated cells carrying out their diverse functions in the organism. Example, secretion, attacking pathogens. Many times a cell will leave the cell cycle temporarily or permanently. It exits cycle at G1 and enters a stage designated G0. Example, G0 are terminally differentiated will never re-enter the cell cycle, but instead will carry out their functions in the organism until they die. G0 can be followed by re-entry into cell cycle. Most of the lymphocytes in human blood are in G0. However, with a proper stimulation such as encountering the appropriate antigen, they can be stimulated to re-enter the cell cycle at G1 and proceed to new rounds of alternating S phase and mitosis. G0 represent not simply the absence of signal for mitosis, but an active repression of the genes needed for mitosis. Cancer cells cannot enter G0 and are destined to repeat the cell cycle indefinitely. S phase or synthesis phase. S phase or synthesis is the phase of the cell cycle when DNA packaged into chromosomes is replicated. This event is an essential aspect of the cell cycle because replication allows for each cell created by cell division to have the same genetic makeup during S phase. A number of events additional to chromosome replication take place. Cell growth continue through S phase, as does the rate of synthesis of a number of proteins and enzymes that are involved in DNA synthesis. Cell growth continues through S phase, as does the rate of synthesis of a number of proteins and enzymes that are involved in DNA synthesis. Once DNA replication is complete, the cell contains twice its normal number of chromosomes and becomes ready to enter the phase called G2. G2 phase or gap 2 phase. Similar to G1, G2 is an intermediate phase, a time for the cell to ensure that it is ready to proceed in the cell cycle, occurring between the ends of DNA replication in S phase and the beginning of cell division. In mitosis, G2 can be thought of as safety gap during which a cell cycle can check to make sure that the entry of its DNA and other intracellular components have been properly duplicated, and to determine if the cell can now proceed to enter M phase, that is mitosis phase, and divide. In addition to acting as a checkpoint along the cell cycle, G2 also represents cell's final chance to grow before it is split into two independent cells during mitosis. Mitosis phase or M phase. Cell growth and protein production stop at this stage in the cell cycle. All of the cell's energy is focused on the complex and orderly division into two similar daughter cells. Mitosis is much shorter than interphase, lasting perhaps only 1 to 2 hours. As in both G1 and G2, there is a checkpoint in the middle of mitosis, that is metaphase checkpoint, that ensure the cell is ready to complete cell division. Actual stage of mitosis can be viewed in animal cell mitosis as shown in figure. Figure shows throughout the interphase the cell is engaged in a growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S and G2. The G1 stage stands for gap 1, the S stage stands for synthesis, and G2 stage stands for gap 2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replication producing two copies of each chromosome. During the G2 phase, the cell continue to prepare for mitosis and cell division. Mitosis or M phase has four stages: prophase, metaphase, anaphase and telophase. During prophase, the chromosome become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called sister chromatic. The nuclear envelope breaks down and spindle fibers form as a microtubule and grow out of the centriole that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attached to each sister chromatid. The centromere that holds sister chromatids together divide and the chromosomes move away from each other along the spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. These are the different phases of mitosis. All organisms are made up of cells which constantly divide and multiply. Cells divide when they need to replace injured or dead cells or as a part of the development of a fertilized egg. 300 million cells are replaced in our bodies every time without our noticing. Do these divisions occur randomly or they regulated? Cell divisions follows a specific pattern called cell cycle. This cycle illustrates the different phases of division. These divisions are regulated and follows a strict chain of events. Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis, nuclear division and cytokinesis or division of the cytoplasm. Cell cycle progression depends on discrete control of points. There are 2 points at which a decision may be taken on whether to proceed through another cell cycle. These checkpoints are like boom barriers on the road. Barriers are lifted only when necessary conditions are met and cell is allowed to progress to next stage of a cell cycle. Commitment to chromosome replication occurs in G1 phase. If conditions to pass a commitment point are satisfied, there is lag period and then the cell enters S phase. This point is most clearly defined in each cell where it is called start. The comparable feature in animal cell is called restriction point. Certain cells can be stimulated to leave G0 and re-enter a cell cycle. Withdrawal of re-entry into the cell cycle can occur only before restriction point in G1. The start checkpoint ensures irreversible cell cycle re-entry even if the conditions later become unfavorable. The physiological factors that control passage through the start checkpoint include external nutrient concentration, presence of mating factors, pheromones, forms of stress and size control. Commitment to mitotic division occurs at the end of G2. If the cell does not divide at this point, it remains in a condition of having twice the normal complement of chromosomes. For animals, cells growing in a culture, G1 control is the major point of decision and G2M control is subsidiary. Cells spend the longest part of their cycle in G1. The length of G1 is adjusted in response to growth conditions. Withdrawal or re-entry into the cell cycle can occur only before the restriction point in G1. When cell proceed past G1, it will barring any accidents complete S phase and proceed through G2 to mitosis, that is entry into cell cycle is irreversible. Cultured cells do not halt in G2. Diploid cell escape those involved in insect embryogenesis, divide and rest in tetraploid states. For haploid cells, it is common for cells to rest in G2 and this offered cell protection in against DNA damage. Since there are 2 copies of DNA in G2 instead of single copy in present in G1. Each cell can enter G1 or G2 as a primary control point. depending on nutritional conditions. Duration of the cell cycle. The length of cell cycle is important because it determines how quickly an organism can multiply. For single-celled organism, this rate determines how quickly the organism can reproduce new, independent organism. For higher order species, the length of the cell cycle determines how long it takes place to replace damaged cells. The duration of cell cycle varies from organism to organism and from cell to cell. Certain fly embryos sport cell cycle that last only 8 minutes per cycle. Some mammals take much longer than that, up to year in a certain liver cells. Generally, however, for fast dividing mammalian cells, the length of cell cycle is approximately 24 hours. Most of the differences in cell cycle duration between species and cells are found in the duration of specific cell cycle phases. DNA replication, for example, generally proceed faster in the simpler the organisms. One reason for this trend is simply that prokaryotes have smaller genomes and not as much DNA to be replicated. Across species and organismal complexity, embryonic cells have an increased need for rapidly in the cell cycle because they need to multiply for the development of the embryo. Early embryonic cell cycles often omit G1 and G2 and quickly proceed through successive round of S phase and mitosis. For these cells, the main concern is not the regulation of cell cycle, but rather in the speed of cell proliferation. As we discussed in the previous section, the length of G1 and G2 vary in cell based on the individual cell's level of preparedness for proceeding in the cell cycle. Remember, cell can enter G0 for extensive amount of time during G1 before continuing on to S phase. If a cell has quickly undergoes sufficient cell growth or DNA replication, the time spent in G1 and G2 will be decreased. It is possible to determine the time a cell spends in a different phases of the cell cycle and its specific location in the cell cycle by feeding cells with the molecules that are only taken into the cell at a specific point in the cell cycle. For example, thymidine is only incorporated into a cell during S phase, and scientists will use thymidine as a tool to mark the onset of S phase. The amount of DNA present in a cell is also a good indication of where a cell stands in the cell cycle. During S phase, DNA is replicated, and as a result, cell in G2 have higher level of cellular DNA than the cell in G1. During interphase, there is visible change in appearance of cells. Synthesis of RNA and proteins occur continuously, but synthesis of DNA occurs only in the discrete period of the S phase, which is shown in the figure. Cell growth is continuous, but the DNA synthesis is not. To summarize this module, we have studied different phases of cell cycle. M phase, that is mitosis phase, which help in cell division. Interphase, which includes G1, G2 and S phase. G1 and G2 are help in a growth of the cell, and S phase, that is synthesis phase, helps in a DNA replication. G1 and G2 also serve as a major checkpoints of the cell cycle. All these phases of cell cycle are helpful in cell growth, cell division and replication of DNA. Thank you.